Hello, everyone, and welcome to today's webinar, Driving Lab Efficiency with PCR Power. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. Please note, we will be distributing poll questions throughout the webinar. Your participation is greatly appreciated. I now present today's speaker, Kyle Hondorp, the Senior Product Manager, Thermocyclers and Plastics, Thermo Fisher Scientific, and Marija Navickaitė, the Product Manager, DNA Polymerase, Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Marija and Kyle, you may now begin your presentations.
Thank you, Sabrina, for the introduction. And welcome everyone to today's webinar, Driving Lab Efficiency with PCR Power. There are three objectives that we aim to achieve with today's webinar. One is help you to discover ways to get the most from your PCR and improve lab efficiency. Two, understand key aspects of critical PCR components and instrumentation. And three, learn how to avoid common PCR mistakes and troubleshoot PCR issues when they arise. Here is the outline of today's webinar. I will start with a brief background about PCR. Then I will talk about ways to enhance your PCR productivity, how to design ideal primers for PCR, and my colleague Kyle will continue talking about instrumentation and plastics for PCR. So let's begin with a brief background. Polymerase chain reaction, or PCR, is a molecular biology method used to amplify or multiply DNA. PCR works even from a single DNA molecule, each amplification cycle consisting of template denaturation, primer annealing, and primer extension steps. During each cycle, each DNA copy is increased twofold. So for example, after the second amplification round, you will have 4 copies of the same DNA. After the 30th cycle, 10 to the power of 9 copies of the same starting DNA. So this is a really powerful technique to study DNA and its analysis. PCR needs several components to work. There is, of course, a DNA polymerase that synthesizes a new strand from a primer by incorporating nucleotides. All these components are mixed in a special plastic plate or tubes, which are then loaded to the thermocycler to begin cycling protocol. And we will review most of these components in more detail in today's webinar. Let's begin from DNA polymerases. A DNA polymerase synthesizes a new DNA strand, and there are several activities that are important for PCR. Synthesis of a new strand relies on 5' to 3' polymerase activity, and all polymerases possess this activity. Another is 3' to 5' exonuclease activity, which removes misincorporated nucleotides. DNA polymerases with this activity are commonly known as proofreading enzymes, because it helps to ensure sequence accuracy during amplification. And these enzymes are commonly used in applications that require high-fidelity PCR. Polymerases possess other activities as well that are important for PCR. Processivity is a measure of a number of nucleotides incorporated by the enzyme in a single binding event until the enzyme dissociates from the template. This activity is also related to the enzyme's affinity or how tightly it binds to the template. High processivity of enzyme contributes to faster DNA synthesis, higher inhibitor tolerance, and ability to amplify longer DNA fragments. Another property, hot start, helps ensure higher specificity and sensitivity. DNA polymerases can be active at room temperature, while polymerases with hot start mechanisms are not. These enzymes are activated during denaturation step at high temperatures, ensuring that amplification begins only in a thermocycler, giving you a specific amplification only. Now let's take a look at how hot start enzymes can help increase your lab productivity. Amplification of non-specific DNA fragments is a common PCR issue. It can be caused by room temperature reaction setup because DNA polymerases are somewhat active at room temperature. This is a problem as primers might bind to non-specific targets and give rise to unwanted PCR products, similar to what you see in lanes 2 and 3 in the gel image on the right. This can be avoided if the reaction is set up on ice, but still enzyme's activity is not fully inhibited. An easy solution to avoid this is to use a hot start enzyme which is modified to be inactive at room temperature. The Hotpot Modifier, it can be antibody, chemical, affibody, or aptamer. In any case, the reaction has to be heated first to activate the enzyme, remove the modifier, and enable synthesis of the new DNA strand. This way, a hot start enzyme provides better specificity and yield in PCR, which can be seen in the 4th and 5th lanes in the gel image on the right. If you are using a proofreading enzyme, blocking 3' to 5' exonuclease activity is especially critical. Non-hot start proofreading enzymes or proofreading enzymes with suboptimal hot start mechanism can have lower sensitivity and specificity, especially when working with high complexity DNA like human genomic DNA. And this is because the exonuclease or the proofreading activity can degrade primers or template even before the PCR starts, and as you can see an example in the image on the right. To prevent this, you can use a proofreading enzyme with stringent hot start mechanism that blocks both polymerase and proofreading activities, and this enables detection of low concentration targets and amplify specific bands. Now, if you set up PCR using liquid handling or robotic platforms, stability of assembled reactions at room temperature is critical to achieve optimal results. Hot start modifications of PCR enzymes makes this benchtop stability possible, and if you use a stringent hot start, you can achieve stability for up to 24 hours. As you can see in the gel image on the right, the same result was obtained when PCR was ran after the setup and when it was left for 24 hours at room temperature. If you are looking for a hot start enzyme, our Invitrogen Platinum PCR enzymes are designed with antibody-based platinum hot star technology. These antibodies fully inhibit enzyme activity until the initial denaturation step, which means greater target yield, sensitivity, benchtop stability, and ability to set up reactions at room temperature. And now let's move on and talk about high-fidelity PCR enzymes. Polymerases can be defined by the error rate that they make. Now, the error rate of an enzyme is defined as the number of incorrect nucleotides per total nucleotides incorporated. It can range from 1 in millions for high-fidelity enzymes to 1 in thousands for low fidelity. If you compare error rates or fidelity information for different DNA polymerases, it is important to keep in mind that there are different ways to measure fidelity, and the results can vary significantly. There are 3 commonly used methods to measure fidelity that are shown here on the slide. So on the left, there is a colony screening assay, Sanger sequencing in the middle, and on the right, next generation sequencing. So when comparing fidelity of different polymerases, it is important to note the method that was used and only compare values that were measured using the same methodology and best if measured under the same conditions. Now, colony screening assays and Sanger sequencing both require huge amount of data to estimate statistically significant and reliable error rates of high-fidelity polymerases. Nowadays, next-generation sequencing enables analysis of large amounts of data and simplifies quantification of statistical data. In this example, in the graph, fidelity of DNA polymerases was measured by next-generation sequencing. The values were normalized to tag DNA polymerase. So here, the value indicates how many times fidelity of the proofreading DNA polymerase is higher compared to tag. Measurements for these polymerases indicated in the graph were made side by side under the same conditions, allowing accurate comparison of the results. And let's look at how high-fidelity enzymes can improve your lab productivity. If you do cloning or sequencing, DNA sequence accuracy is critical. You want to minimize possibility of having an error in the PCR sequence, and this is where it is critical to use a high-fidelity polymerase. If you are to choose a PCR enzyme with the highest fidelity possible, that will increase your confidence in the DNA sequence accuracy after amplification and save your time from troubleshooting and repetition of the experiment.
Displayed on your screen is our first poll question. What kind of issue have you experienced while performing PCR? A, incomplete or no amplification. B, poor sensitivity and or specificity, errors in PCR amplicons. C, contamination, or D, other.
We will now look into PCR enzymes with high processivity and how it can simplify PCR when working with challenging samples. As was mentioned before, processivity is defined as the number of nucleotides incorporated by the enzyme in a single binding event to the DNA template. The higher the processivity, the more nucleotides the enzyme can incorporate without falling off the strand. This often leads to faster synthesis rate and increased enzyme's affinity to the substrate. So how can a higher processivity of an enzyme help in a daily PCR? If you amplify DNA from inhibitor-rich samples like plants, blood, or soil, you may have difficulty with your PCR. This is typical because PCR inhibitors are not completely removed during DNA purification. In such case, if you use highly processive enzyme, you can overcome PCR inhibition because these polymerases tolerate suboptimal reaction conditions. You can see an example here shown on the right where DNA samples were amplified with spiked known PCR inhibitors using highly processive platinum 2-tack DNA polymerase. It is often a challenge to amplify long DNA fragments, for example, longer than 10 kilobases. Success rate is often low, there is high error rate, and long cycling protocol. In such situations, high-fidelity enzyme with high processivity can simplify the workflow because the enzyme ensures low error rate and better amplification of longer fragments, while high processivity of the enzyme leads to shorter cycling time and also increases success rate of the amplification overall. The gel image on the right side shows amplification of 20 kilobases from human genomic DNA and even up to 40 kilobases from E. coli genomic DNA using our high-fidelity platinum SuperFi 2 DNA polymerase. GC-rich DNA amplification is another common challenge in PCR. GC-rich regions are harder to separate and they can form secondary structures after the separation. Most often, additives like DMSO are used to overcome these challenges. Also, there are specialized enhancers available with some enzymes that simplify reaction optimization. Now, our latest platinum SuperFi 2 enzyme comes with an innovative buffer that allows amplification of GC-rich targets without a need for an enhancer, bringing down all the optimization efforts to a minimum. And in the next section, I will talk about some other ways how to increase efficiency when doing PCR. And one way is to choose direct PCR approach, which enables PCR without prior DNA purification, cutting down the workflow time significantly. Conventional PCR involves DNA purification, which can take from an hour to several hours, depending on the method. Direct PCR does not require this, and you can get from sample to PCR in less than an hour. In addition, direct PCR is handy when working with very small samples, as it requires minimal handling and minimizes chances to lose the sample completely. It is also an ideal choice when working with many samples in high throughput labs, for example, for genotyping experiments.
Displayed on your screen now will be a second poll question. Would you like to receive a quote at an early stage of your research? And for our third and last poll question, would you like to be contacted by our sales support team to help you make a purchase?
Now, the annealing temperature for primers often needs optimization to help ensure efficient binding of the primers to achieve the best result. It is a long and tedious process, especially if you work with multiple primer sets. However, our latest platinum PCR enzymes are formulated with universal primer annealing temperature. These enzymes allow you to use 60 degrees annealing temperature for different primer sets. You can see an example on the right where all DNA fragments were amplified at 60 degrees, while their actual calculated melting temperatures ranges from 59 to 69 degrees. This universal annealing feature provides two additional benefits shown here. First, you can amplify multiple different length fragments in the same run using the same 60 degrees annealing temperature and using extension time of the longest fragment as depicted in the image on the top. Fragments up to 5 kilobases can be amplified without losing specificity or yield. What this also means is that different PCR assays can be cycled together using one thermocycler as shown in the bottom image. Typically, with other enzymes, different PCR assays need to be run sequentially or on different instruments. So really, universal primer annealing significantly saves time when it comes to reaction preparation and also a cycling step. And lastly, I will just briefly summarize how to choose a right enzyme based on your application. If you need to detect a target sequence, just basically get a yes or no answer. Use a low-fidelity enzyme, for example, platinum 2-DNA polymerase. Now, if you need to preserve DNA sequence accuracy in applications like cloning, sequencing, or mutagenesis, high-fidelity enzymes like platinum SuperFi 2 will benefit you. If you want to detect target DNA from samples without purifying DNA, choose platinum direct PCR universal master mix, because that will save you a lot of your time. And down at the bottom, there is a link where you can find more information about our platinum enzyme family. And now primers. Primers are essential for PCR. So let's look at some tips how to simplify your PCR by designing good primers. If primers were not designed correctly, you will either get no amplification or lots of non-specific products. It is essential to follow primer design rules to design primer for optimal results. Here on the right, you can see several tips to keep in mind when designing primers. That is a primer length, GC content, number of dinucleotides or repetitive bases. These are just some of the aspects to consider when designing a new primer pair. Or you can use specialized tools for primer design where you do not really need to think about these rules. And one example would be OligoPerfect Primer Designer online tool. And now my colleague Kyle will continue talking about thermocyclers and plastics.
Thank you very much, Marija, for all that wonderful information. Beyond the selection of appropriate reagents and primers to improve PCR efficiency, instrumentation also plays a critical role in driving PCR success. It is important to select a thermocycler that ensures precise temperature control. Thermocyclers that deliver inconsistent temperatures can impact the accuracy of PCR results, especially if seeking a specific annealing temperature or performing PCR optimization protocols. This can lead to confusing results and require additional time to re-optimize protocols to overcome this. One way to achieve the solution is to look for a thermocycler that provides precise temperature control. A traditional gradient temperature control allows you to set a minimum temperature on one side of the block and a maximum temperature on the other side. Heat then transfers across the block in a gradient, but it lacks temperature accuracy at any specific well. There are technologies available that allow for precise temperature control. The VeriFlex block technology, temperature control technology, utilizes blocks constructed of metal segments. Each segment contains its own heating and cooling elements. Depending on the instruments, you can find either 3 or 6 independent temperature zones. With the ability to uniquely control each zone, you're able to achieve accurate temperature control, which is ideal for enzyme studies, restriction digest, or any workflow requiring temperature precision. If your lab is limited with the number of thermocyclers available, this can create backlogs in scheduling and delays in time to results. Today, there are thermocyclers with very basic features and small footprints available at a reasonable price. This makes it easy to obtain personalized instrumentation, allowing more lab members to have access to their own benchtop thermocyclers. One of the challenges many researchers face when switching to a new model of thermocycler is the ability to transition protocols to the new instrument without needing to re-optimize experiments. Thermocyclers can operate using different ramp rates. This makes it challenging to replicate experimental results across instruments. To solve for this, look for thermocyclers that offer simulation mode. Simulation mode provides a library of instrument modes that mimic the ramp rate of those instruments. This makes it easy to transition protocols and workflows without the need to re-optimize. Simulation mode is available on Applied Biosystems thermocyclers. Another way to improve PCR efficiency is having the ability to manage PCR protocols and runs while away from the lab. Look for instruments that offer secure remote access with a Wi-Fi or cloud-enabled instrument. Securely connect through your mobile device or desktop computer to start or stop a run, design and share protocols, or export data. This gives you greater control over your research, whether you are in the lab or working remotely. One area to improve PCR efficiency that is often overlooked is the selection of optimal PCR plastics to fit your instruments. Thermocycler manufacturers design thermal blocks and instrument lids to fit either a limited or wide range of PCR plastics. Plastics that don't fit the thermal block can lead to deformation or crushing, causing sample evaporation or loss of results. To solve this, use the manufacturer-recommended plastics designed to fit your PCR or qPCR instrument. You can find recommended plastics by using manufacturer compatibility tables or use free online PCR plastic selection guides to confirm the fit of the plastic with your specific instrument model. At the bottom of the slide, you will find a link to an online plastic selection guide for plastic compatibility with Applied Biosystems and other instrument manufacturers. PCR tubes are available in a variety of sizes and formats. How do you select the right fit? First, you need to know the profile of your thermal block. Are you working with a standard 0.2 mil block or a 0.1 mil, also known as a fast thermal block? The tubes are different sizes to fit the appropriate block style. So make sure you select and know the block format that you're working with for the best fit. The second consideration is what style cap do you need? Domed caps are used in endpoint PCR and can help absorb lid pressure and prevent tube crushing in instruments with inner concave lids. Flat caps are recommended for instruments that have a flat lid. And ultra clear caps are used when working with real-time PCR instruments. For experiments that require additional throughput, we recommend the use of tube strips. Tube strips are available with either 8 tubes or 12 tubes. You can find tube strips that come with tubes and caps separately, or to prevent cross-contamination, we recommend using tube strips with attached caps. This will allow you to independently open and close each tube as you're working with that particular sample. Again, use compatibility tables or online selection guides to make sure that the plastic is the right fit for your thermal cycler. Ccr plate selection also depends on the instrument. Plates come in a variety of styles. They can be non-skirted, semi-skirted, or also known as half-skirted, or full-skirted. Automation usually requires a semi or full-skirted plate, while non-skirted plates offer more flexibility to fit many thermal block formats. The Applied Biosystems thermal cyclers use semi-skirted plates. Deck style is another consideration. A flat deck offers the most universal fit with instrument lids and is also compatible with automated heat sealers. While raised decks are plates that have a plastic lip that goes all the way around the outer edge of the plate. This is required to help balance lid pressure on certain thermal cyclers and is required to fit with certain thermal cycler lids. Finally, when should you seal with caps versus films? Cap strips can be used for both tubes and plates. They must fit securely and seal tightly in order to avoid evaporation. Film is often used when working with 96 or 384 well plates, and film has the advantage that it can be cut into smaller sizes to fit segmented plates containing 24, 32, or 48 wells. Adhesive clear film or adhesive foil can be used in endpoint PCR, while optically clear film is recommended for real-time instruments. Optically clear film is a pressure-sensitive film and requires pressure to be applied to form a proper seal. For more information about forming a proper seal, we do recommend the use of an adhesive film applicator. When using the applicator, you apply pressure gently and evenly across the rims of every well to ensure that the adhesive seals with the well to prevent any sample loss. Other sealing considerations may include considering the deck of the plate, especially if you're wanting to use an automated sealing method system, or the type of lettering. Do you need raised or engraved lettering to ensure you get a proper seal? For more information about any of the PCR workflow topics we have discussed, we have assembled some resources. We have a PCR workflow solution brochure that goes through various portions of the PCR workflow. You can also find more information on our product pages for our Pcr Enzymes, thermocyclers, or Pcr Plastics. You can click the link to get access to our PCR application notes. We also have a PCR education webpage that will discuss considerations for PCR as well as common troubleshooting tips. Links to our various brochures and handbooks will help further your understanding of the PCR workflow and the products available to support your research. Our hope is that this webinar provided you with helpful insight to drive your lab efficiency with powerful PCR tools designed for speed, simplicity, and accuracy. I thank you for your time today, and now Marija and I will be happy to take any questions.
Thank you, Marija and Kyle, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Let's get started. Our first question: do I still need to use the universal annealing temperature? I trust annealing temperatures I calculated.
Oh, thank you for the first question. The answer is no, you don't have to use universal primer annealing temperature. The enzymes will work with the calculated temperatures as well, but the universal annealing feature is convenient when you are working with multiple primer sets.
Wonderful. The next question I have: how do I get samples of the enzymes you mentioned?
Thank you for the question. Yes, so to get the samples, please contact our Thermo Fisher sales representatives. And in some countries, you can also request product samples directly on our website.
Wonderful. The next question I have: what type of PCR plastics do you recommend if I am automating some of my workflow with a robot?
That's a great question. There are actually two factors to consider when thinking about using plastics for robotics. The first one will be the skirt type. As I mentioned before, semi-skirted and fully skirted plastics are usually recommended for automation. That's important because the robotic grippers or paddles that move the plates around will need some surface in order to do their gripping. The second consideration is the PCR plastic material. Some plates are prone to warping and cannot work well within automated processes. Therefore, we would recommend plates that are constructed with a stronger polycarbonate frame, which resists distortion caused by those robotic grippers and tolerate the robotic transfer well. Those plates still contain the same thin-walled polypropylene wells that are traditional in all PCR plastics, but that frame makes an important difference. And for Thermo Fisher Scientific, we have both the Endura plate and the Armadillo options for polycarbonate frame plastics.
Thank you so much. The next one I have: do your thermal cyclers have gradient capability?
Great question. The ProFlex, Verity Pro, and SimpliAmp thermocyclers feature our VeriFlex block technology. So this is that precise temperature control. The instrument either contains 3 or 6 segmented metal blocks, each with their own heating and cooling element, so that each zone can be set with a unique and independent temperature. Our Verity Pro instrument also has the ability to set temperature based on the minimum and maximum temperature across the block, or to set the midpoint, which is our zone 3 range of the thermal block.
Thank you so much. The next one I have, fidelity value that you showed in the graph does look different from what I see on my enzymes manual. Can you explain that?
That's a great question. Thank you. Yes, correct. Fidelity values can differ depending on the methods used in measuring, and so different companies will show different values for their products. As it was mentioned during the webinar, for example, if values were measured using the lac-based method, it will vary significantly compared to values measured using next-generation sequencing. And this is especially true for higher fidelity enzymes, where you really need to use a sensitive method to measure fidelity accurately. The bottom line, though, is when comparing fidelity values of different enzymes, they should be measured side by side using the same method under the same conditions.
Wonderful, thank you. We have time for a few more here. Do you have direct kit for RNA?
Yes, we do have a direct kit for RNA. We recently launched a new kit. It is called Superscript for Cells Direct. It synthesizes the cDNA directly from RNA of cell samples.
Thank you. Another question. Tell me more about wireless connectivity.
Yes, cloud-enabled instruments allow you the freedom to design and securely upload your methods, monitor runs, or check instrument availability from any mobile device or desktop computer. You simply need to look for an instrument that's Wi-Fi or cloud-enabled and connect to it.
Great. And how can I prevent sample evaporation during PCR?
Great question. Proper sealing of your PCR reactions is essential to preventing evaporation and sample loss. If you're using an adhesive film, you want to make sure that it properly fits the size of the plate and to align the cover across all wells. Adhesive film will stick. An optical film does require, it's pressure sensitive and requires pressure. But in both cases, we do recommend the use of the adhesive applicator tool to firmly press across the entire film and to make sure that the rims of every well are securely connected and sealed. You can also use cap strips with plates. So if you're not getting a proper seal, that's another option, is to consider using cap strips, as long as you ensure that the cap strip has a tight fit in with the plate. So those are a few suggestions on how to properly seal.
Thank you. Okay, let's see here. The next one I have. Can the choice of hot start modifier make a difference?
Oh, that's a great question. Thank you. Now, my short answer would be yes, there is a difference, and it mainly depends on your application. So, for example, antibody modifiers are quite stringent and can fully inactivate the enzymes, but antibodies are proteins, so they may not be suitable if your application requires low protein concentrations. Chemical modification modifies the enzyme. So the enzyme that has a chemical hot start may not be fully reactivated during the activation step, which quite often impacts the amplification of log sequences and sometimes enzyme sensitivity. And then antibody and aptamer. So these are small molecules. They're not proteins, but they may not be as stringent depending on their dissociation temperatures.
Thank you so much. And the next one I have, direct PCR sounds great, but what are its drawbacks?
Oh, it's a good question. So one thing to keep in mind with direct PCR is that you are not actually purifying DNA, which is good if you don't need it, but if you need a purified genomic DNA for a certain downstream application, then direct PCR may not be a good approach for you to use. The next thing is that sample size is important because if you add too little, you may not get any amplification, the signal will be too low, or if you add too much of your sample, it will inhibit the PCR reaction. We do have some guidelines on our website, how much of samples to add depending on the sample type and depending on the protocols used for direct PCR.
Wonderful. I have two more questions here. Premier design rules look complicated. I've seen some tools on the web. Any that you would recommend?
Yes, of course. We recommend using our free online tool called OligoPerfect Designer. The design rules are built into the algorithm, so you don't really need to think about all those rules. And we also recently updated its bulk ordering function to simplify ordering if you have many primer pairs.
Perfect. And I have one more question that we're going to wrap up with. Do I need to measure fidelity of the enzyme that I am using?
No, you don't have to measure fidelity of the enzyme that you are using. It is a really tedious process, and usually the enzyme suppliers do that for you.
Wonderful. Thank you again, Marija and Kyle, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you again, Marija and Kyle. Goodbye.